So the first lecture today is Introduction to Metabolomics. Sort of what is it? Oops. And I think that the best way to think about metabolomics is in the context of essentially systems biology, which I'm going to be talking about and defining a little bit more as we go. And everything starts in biology with genomic with DNA, genome sequences, and natural genetic variation. So things, all of our DNAs have natural variation, and that forms the basis of pretty much everything going downstream from that. So DNA is modified, as you know, epigenetically, so that different genes are activated or repressed, and that's an area that you can study on an omics level. The transcriptome are all of the expressed genes. We can study that on an omics level. Proteins are made from transcripts, again, proteomics. And then finally, metabolomics. And I think it's often said, and I think it's true, that metabolites are really closest to the phenotypic variation that you're trying to look at in a study. There are a lot of reasons for this, but one of the biggest ones is they feed back on everything, all other layers of information in systems biology. Essentially, each one of these ohms have feedbacks from the metabolome, and the metabolites, as we'll discuss, change very rapidly in response to the environmental circumstances, and so they're really a good readout of what's going on. So all of these, as I indicate here, are influenced by the environment, and so those are the kind of things that we might want to study in terms of, disease or disease and response to treatments or exposure to various chemicals. Like PFAS is a popular area of study these days, for example, and to try to study what happens with PFAS or infectious disease. So anyway, this is the basis that we want to be thinking about metabolomics and natural and systems biology. So the just another little different way of looking at it. So systems biology is really sort of the interdisciplinary study of complex interactions that are taking place in biological systems. It's based on, my definition at least, of systems biology is that it's based on these omics. So we're talking about metabolomics in this workshop, and one part of metabolomics, I should stress, and NMR metabolomics. But then mathematical models are really necessary to bridge all of these things together. So that's where the systems part of systems biology really comes in. Genomics are the study of genes. It's the only ohm that's not really context dependent. You're sort of born with your genes. You do get mutations along the way, but it's not so much context dependent. Transcriptomics is I'm sure all of you are aware. It's very context dependent. You can get a lot of information out of transcriptomics by looking at different conditions, different genotypes, and so on. But it's all the messenger RNA in cell, tissue, or organism. Proteomics, all the proteins in a cell, tissue, organism, and metabolomics. Or sometimes you hear the word metabonomics, which I just use interchangeably. There are people out there, especially Jeremy Nicholson's lab, that really make a distinction between these. But for the purposes of this workshop, we'll use them interchangeably, and usually metabolomics. So in metabolomics, we refer to all the metabolites in a cell, tissue, or organism. And so you can think of DNA as like the ultimate potential in a cell, what's possible, and RNA, you might think of this almost like the direction that it's going, what appears to be happening in a cell at any one moment. The proteins, what functional capabilities are in a cell. And then the metabolites are often referred to as a limiting currency of a cell. What's actually happening at that moment? And so that's what makes metabolomics so versatile and so valuable, is this concept that they really are sort of the limiting currency of what's going on in physiology and on a day-to-day basis. And so the thing that I've come to really like to use as an example of this is ATP so this is Usain Bolt going in a 100 meter dash, and the question is, how long does ATP last in a sprint? If you want, you can put some answers into the chat. I'll be curious to see kind of the range of how long. And I guess I'm going to ask another question in the chat that's not on this slide, but it's related. And that's, how much ATP do any of us make in a day, let's say? So put some answers in chat. I'll be curious about that, and then I'll tell you what the answer is in a minute. You can Google it too if you want, or you can just put it out there. So how long does the ATP for Usain Bolt, for example, last in a sprint, and how much ATP do all of us make in a day? That's what I want you to think about a little bit. So, and then it's going to be different about whether, if you're in a sprint or a marathon. So clearly the ATP that's going to propel a 100-meter dash champion over the finish line is not going to be the same energetics as what propels a marathon runner over the finish line. So we have to look at this differently. This is some basic biochemistry from one of my favorite textbooks, Vote and Vote, where they compare it. This is just looking at basically ATP systems. So if for anaerobic systems, so sprints, powerlifting, tennis serve, shot put, these kind of things. And I guess sprints and football line play are put down here for phosphocreatine, but basically anaerobic systems have just a tiny bit of we only have a few seconds, so it's really less. It's less than it's less than 5 seconds for sure, and even really less than 3 or 4 seconds of ATP on board as ATP that's ready to be hydrolyzed for activity like a sprint. Then there's this phosphocreatine that is rapidly converted into ATP. That is a very fast transformation, and there's a little bit less than 10 seconds of this on board for a sprint. At that point, the only way that energy is made again is going through glycolysis and anaerobic glycolysis, where you can rapidly make ATP but not very efficiently make ATP. And this is the sort of this is where an intermediate length race, a swim or a 400-meter race, which are really fast, but not as fast as a 100-meter dash. So this is where the ATP is coming from, the anaerobic glycolysis at that point. And then anything that's longer than that, like the marathon races, they really have to go through oxidative phosphorylation. So the TCA cycle has to become engaged, and then this will make a lot of ATP, but much more slowly than the ATP that's available early on. And so I'm curious about whether did anybody make a guess about how much ATP we make each day? Anybody want to just be brave and unmute and say what you think? Going once, going twice. Well, it's about our body weight every single day of ATP that we make, every one of us. So it's really a remarkable amount of ATP that the human body makes every day. So that I use this as an example. It's probably one of the more dramatic examples of the limiting currency of metabolites, but I think it's a relevant one, and it really illustrates the importance of that concept. I love this because I've been thinking about this in the terms of teaching metabolism for several years in terms of the ATP for a race. And then in 2021, during the Tokyo Olympics, the New York Times created some really beautiful graphics of how that they asked this question: how fast the Jamaican sprinters who finished 1, 2, and 3 in the women's 100 meters race did that, how fast they ran these races. And it was really interesting. So they showed the starting line early in the race and then a little bit into the race, and they basically plotted the plotted right here distance on the x-axis, so 0 to 100 meters. These are 100-meter dashes, and the vertical axis is the speed of each runner at every point along the way. So you can see the very rapid acceleration that these athletes have early in the race, and then they plateau off at almost 25 miles an hour in this middle stretch of the race. And the thing that really caught my eye is this end of the race where speed starts to drop off. And you notice, so the 100-meter dash, let's say it's roughly 10 seconds or so. So in terms of the numbers that we just talked about, so ATP a couple of seconds, phosphocreatine, it'll go out to just a little bit less than 10 seconds. And so I think that we're seeing reflected in their speed the effects of. So here's the last bit when they're just running short on ATP and switching over to glycolysis, anaerobic glycolysis, but that's about as much as they could get out of their body. So I think that this plot showing how their velocity is changing over time is really instructive in this sense. So I loved seeing that. Okay, so let's dig into metabolites a little bit more in general, not just for 100-meter dashes and such. And one question that you could ask is, how many metabolites are there? A lot of people do ask this question. This is a KEGG representation, the Kyoto database for metabolites representation of you see this a lot because it's such a pretty slide. But you have all of these different things: glycans, lipids, carbohydrates, amino acids, TCA cycle, energy, energy and TCA, secondary metabolites, all of these things that form us and are part of this very complicated map. And you can go to KEGG and get an answer of how many metabolites in there. But the fact of the matter is that there are a whole bunch of compounds that don't seem to fit into the KEGG map. And we collectively refer to this as metabolic dark matter, following on the dark matter of the universe and such. So I'm just representing this part right here as the known universe of metabolites. And then outside of that is meant to represent the things that we really don't know, and there are a lot of reasons that we don't necessarily know them. It could be because microbiome. So I'm sure that you're all aware of the fact that we're not alone. As humans, we have lots and lots of microbes in our guts, many more cells than our own cells, and these are actually helping us to live. I mean, they're beneficial. They're helping us digest and do our own metabolism. And so they also contribute in a significant sense to our overall metabolic health. The exposome, so anything that we are exposed to in the environment, whether it be in the water, the air, the food that we eat, drugs that we take, but I think that you're probably all familiar with polyfluorinated alkyl substances, or PFAS. This is present in much of the drinking water that we're exposed to. Most people in the world, but certainly in the U.S., have measurable amounts of PFAS in their blood. This bioaccumulates. It's one of these real concerns about what is it doing to us. There are a lot of different things that are out there. PFAS is one of the more popular these days. There are actually repair pathways that are understudied, but known, there are a lot of, if you look at, if you study metabolism, you'll know that there are many intermediates along metabolism where there's some pretty nasty intermediate chemicals that are made, that, free radicals or charged molecules that would cause problems. And but these, especially the free radicals, really can cause some damage if they escape in from the active site of enzymes. And sometimes things don't always go well. I think many of you are familiar with DNA repair, the concept of DNA repair, and that happens. Basically, none of us would be alive if it weren't for the fact that our DNA, our genome, is being repaired constantly when we're exposed to things like sunlight. But the same thing happens with metabolites, but we just don't know as much about it as we do for DNA repair. So if metabolites get out of pathways, they can kill cells, and so there are pathways that are activated in response to that. And so the bottom line is that these are at least three major areas that impact the list of KEGG metabolites or other database metabolites, and impact how many we actually can see in a metabolomics experiment. Okay, so we really don't know. David Wishart, who he created the Human Metabolome Database. I'll talk about that a little bit later in this slide set. He estimates a lower limit, and my guess is that this has probably changed since I spoke to him at one recently, a few years ago, about this, but a lower limit of 150,000 metabolites in humans, but estimates it could be much greater. I'll talk about numbers a little bit more on the on when I get to the HMDB in a few slides. So there are a lot of metabolites. We don't know how many there really are, and I think it depends on the context and what people are exposed to, what microbes are in us, et cetera, et cetera. Okay, so in metabolomics, there are two main ways, a bunch of other ways, but two main ways that people use to measure metabolites. I should just add right here that there are some, like one of the most interesting alternatives that I'm not showing here, and we're not going to be talking about in this presentation, are like Raman and infrared measurements. You can do some really detailed measurements on whole cells using these technologies. So I think that they're emerging areas, but most of the studies that you come across today either use mass spectrometry or nuclear magnetic resonance. And as you know, this workshop is focused on NMR. I'll start with what many of you probably already know about for mass spec, that the reason mass spec is really, I would say, the dominant measurement tool for metabolomics these days, for a number of good reasons, but I'm going to tell you why I think that NMR needs to have a lot of attention paid to it as well. Mass spec is really sensitive. It's much more sensitive on a molar level than NMR. It can go much lower concentrations than nanomolar, so you can get more coverage of interesting low concentration metabolites than you can with NMR analysis. You can look at many thousands of compounds, and every time I see this word compound in here, I'm reminded that I really should change that to feature, but because it is hard to identify a peak in a mass spectrum and assign it with confidence to a known compound. But you can measure a whole bunch of peaks, and I'll talk about that a little bit more in the next slide. Mass spec is usually used in tandem with liquid chromatography, gas chromatography, and MALDI. These are different ways of ionizing compounds, and they have additional information. And actually, on Thursday of this workshop, we're going to be talking about putting liquid chromatography as part of NMR, because I think that the separation that is inherent in a lot of LCMS experiments and GCMS experiments is really important to the success of mass spectrometry. One of the problems with mass spec is that it really is difficult to quantify compounds. If you want to get absolute quantification, you really have to have standards. ion suppression can be a problem when compounds come out at the same time. So even getting relative quantification can be a struggle. You might think that you're selecting for something for MSMS, but you can get a couple of different things leading to some misunderstanding in mass MSMS spectra. And then one of the most important is that mass spec, it doesn't use a lot of material, but it's destructive, so you can't get it back. So now the good parts about NMR, and the reason why we really think that NMR has a lot of value to bring to metabolomics. First of all, it's quantitative. The technique is quantitative, and I think we often, well, we like to say that, but it's hard to do. And actually, one of the methods that you're going to hear about tomorrow, called SAND, is meant to make this quantitative nature a lot easier than it currently is. So we have a lot of information that we can get out of NMR. It's currently very difficult in metabolomics. It's really important for structural identification. There's we have limited amounts of information in metabolomics, and mass spec. If you get an M over Z or an MSMS fragmentation pattern, NMR, you can get NMR spectra, you can get 2D spectra, but the 2D experiments, especially the kind that you'll see more of on the last day when we talk about C13 NMR, those are really helpful for structural identification. You can analyze tens of hundreds of compounds, and there's no requirement for ionization. So I think that even though NMR doesn't see as many features, we can see a bunch of compounds and oftentimes get the high points of the metabolic pathway. It's highly reproducible compared, especially to mass spec and chromatography, and it's non-destructive. So that non-destructive nature will emphasize some of this in the fraction library work that we talk about on Thursday. It's not as sensitive, so you'll have fewer compounds detected, but it's not bad. So you can usually get in a low micromolar, even high nanomoles of material using instruments such as the 800 megahertz with a small volume probe that we have in our facility, and some of you will be using today even. 2D and 1D experiments can take longer time than others, and then overlapping signals really are a problem. And so this overlapping signals is really what much of this workshop is meant to address. Okay, so I've mentioned the large number of features that that you can measure in an LCMS experiment, and I want to just bring to your attention a paper that I think is important from a friend of mine, Gary Patti, that he published a few years ago, and he still is doing a lot of really outstanding work in this area and has taken this even further. But what Gary did was he took a metabolomics data set of E. coli, so very well-known organism, and he started off by just measuring E. coli as a mass spec person would do, and he could get 25,000 features, so tons and tons of peaks that he could measure out of the mass spec. But then in this paper, he describes what he does to what the term that he uses is credentialing. So he will take these 25, subtract off isotopes, adducts, artifacts, contaminants, things that mass spec people know that you should do. But then he does a lot of other work where he labels the organism with C13. He requires that metabolites are biosynthesized. And the bottom line with this is that 25,000 features ended up with much less than 1,000 unique metabolites. And so this paper created some controversy because a lot of people misinterpreted it by saying, that Gary Patti was saying that E. coli only has 1,000 metabolites, and that's not what he's saying. What he's saying is that out of those 25,000 features, I only can find about 1,000 metabolites. But so anyway, the ratio of what you can measure in a mass spec compared to what's actually there is lower than you might think. And so I think it it's maybe not that far away from NMR in terms of the number of compounds that we can see. So going on to the metabolome, all of these, the metabolome is sensitive to all of these things. So genotype, changes in messenger RNA, changes in protein, microbes, and external environment, disease, diet, treatment. These are all of the things that make it so that we can actually that make people want to do metabolomics. You can study disease and get funding from the National Institutes of Health and understand how diseased people are responding to treatment, for example. This sensitivity is really it's both good and bad because we have to be very careful about cause and effect and also in how we prepare our studies because all of these things will have an impact. And so it really emphasizes the importance of a good study design and knowing what you're really measuring in a study. And unfortunately, we don't have a lot of time to talk about that in this workshop in any great detail. But I do also want to point out, so we like to think about you probably have seen this figure if you've been in the metabolomics area for a little bit, but I think it's a really interesting one. We like to think that that metabolomics is the newest kid on the block in omics. I mean, it really has come after genomics and any sort of DNA, RNA sequencing came first. Proteomics really started to grow a lot before metabolomics. And so we tend to think of it as new, but the concepts have been around for quite some time, as I'll emphasize here and on a few other examples going forward. And this is a figure that Jeremy Nicholson published a few years ago in 2008, where he showed this reproduction of a physician's wheel that Pinder published to explain how a physician should diagnose disease based on the color, taste, and smell of a human's urine. So they could actually each one of these is a category of disease that a person might have based on those properties. And it's not as gross, but it's not as crazy as you might think, because if any of you are diabetic, you probably know that your urine can become sweet, and that you see a lot actually in a metabolomics measurement. You can see a lot of glucose in urine, and so this is one of the it has a lot of foundation to it. I also like to give the example of asparagus, at least for me, and I think that well over half of you, if the statistics work like they are, they should be. Basically, if you eat asparagus, you get this kind of funny smell to your urine right away, right after you eat it. So you eat it, and if you need to go to the bathroom, you could, even 10, 15 minutes after you eat it, you can smell things. And these things that you're smelling are various sulfur-containing compounds that have been identified and characterized. And some of these references here, we'll go over them. This has been known for some time as well. So here's something from Marcel Proust in 1913, that asparagus transforms my chamber pot into a flask of perfume. So people have known about that what you eat ends up in your urine, and so that gives hints about the information that's out there in things like human urine. This is what human urine looks like on a 600 megahertz NMR spectrometer. There are a lot of peaks in here and a lot of detail. And I should just say that this workshop is really one of the main points of this workshop is to ask the question, how do we deal with all of this stuff? And that's what we're focusing on, is sort of three different approaches to how do you deal with this extreme amount of overlap that we see. I give up here a reference to a figure by Bautor et al. This was the human urine metabolomics. This is from Wishart's lab, so it's the human urine metabolome paper that they published several years ago now. And the reason I like to show this is that actually NMR was able to detect most of the compounds, more compounds, unique compounds than any of the other kind of mass spec or other type technologies in this case. So NMR is particularly good at human urine in terms of measuring lots of features. Going back a little bit more to the history of metabolomics, Linus Pauling was one of the first people in the modern age after Pindar, a long time after Pindar, but Linus Pauling, one of the greatest scientists that ever lived, hypothesized that you could actually predict human health based on profiling body fluids, and he was really the forerunner of the concept of using things like human urine to diagnose disease. So chromatographic separation was developed in the 60s, and Robinson and Pauling published this quantitative analysis of urine vapor and breath by gas liquid chromatography in 71. And then also the metabolome and metabolomics concepts were proposed in the 1990s as a term that came way after Pauling was thinking about this. I'm going to show some data from Pauling in a minute. And then just to finish off in terms of this timeline sort of history, the David Wishart's Human Metabolome Database made its first release in 2007, and it had 2,500 metabolites, 1,200 drugs, and 3,500 food components. The current release of HMDB has a lot of different categories. And when I looked just a few days ago, around 220 or 221,000 compounds are in there. So that's a good resource to know about. This is from this Pauling and Corey and others work. They were able to take this is urine put onto a gas chromatography instrument. So this is not GCMS. This is just gas chromatography and detecting peaks as they come off. And this is just a long trace going from 0 out to the end. And they were able to actually quantify 280 different substances from this compound. And that's where this idea came about, that maybe we could use that for profiling and characterizing human disease. So finally, what is a metabolite? We often say that metabolites are less than 1,000 or less than 1,500 Daltons. That's always pretty movable. It's not really, I don't think that a molecular weight cutoff is necessarily all that useful. There are a whole bunch of things that qualify in my book as metabolites, peptides, microRNAs, sugars, all of these things that are in the KEGG chart, drugs, xenobiotics, exposome. So it's a complicated mix of a whole bunch of different things out there. So the last, then they're also very diverse, making it a technical challenge. That's one of the problems with it, is that they're so chemically diverse. So the last bit, I just want to emphasize the overall workflow. So you have to start a metabolomics study with a concept of an experimental design. And this really should, I recommend this to everybody all the time, that you should start with a statistician early in the process, just so that you have an idea of what you're really trying to measure. Is your study design suitable to get the information that you need to realize what you're trying to find, to test your hypotheses? And then you have to go through and work hard on sample preps that are going to give you what you want. You have to make measurements that will give you what you want. You have to do data processing to extract information. There's variation in each of these steps, including the statistical analysis and data processing. And so all of these steps are important. And the thing that I we have to limit what we present in a short workshop, and what we're really limiting this workshop to are measurements that you can do on NMR and some of the data processing steps that we find most useful in our lab. But that's not to imply that experimental design and sample prep and such are not useful. But this is what we're going to be focused on. And then this is the last slide, and then I'm going to switch slide decks to NMR. I do want to also reinforce the fact that we want our data to adhere by these so-called FAIR principles: findable, accessible, interoperable, reusable. This is really important to reproducibility in science in general, but especially in omics. And for example, the kind of resources that you heard yesterday from Mark describing what we're trying to do in NAN and NMR box and BMRB, we're really dedicated to fair data principles. And I hope that you will be too in your research.